Hello, Tarja Malm. You are a professor of molecular neurobiology and the research group leader of the Neuroinflammation research group at the University of Eastern Finland. Could you briefly introduce your research group and some of its key focus areas?
Sure. So my research group consists of approximately 25 people. We have equal number of postdocs and PhD students, and then 4 technicians. Our group is heavily focused on human-based modeling of brain cell functions. We intensively develop human-induced pluripotent stem cell-derived model with a special focus in developing organoid models that enable investigation of microglia-neuron interaction during developmental phases of the brain. And we also utilize a lot of human brain samples that we received from the brain surgeries at the Kuopio University Hospital. And we use these samples for interrogation of human cellular functions and states in early Alzheimer's disease, as well as epilepsy.
Wow.
And in all of our studies, we combine interdisciplinary approaches to link the cellular states to neuronal operational properties.
Wow. And what are some of the key issues or research questions that your group is tackling?
So overall, my group aims to understand how and why microglia become malfunctional in different brain diseases, and how those malfunctional microglia impact neuronal network functions. And for example, one of the key questions we're trying to answer is how early AD or Alzheimer's disease-related pathology alters single neuron and neuronal network operational properties. And for this, we use brain biopsies that we get from idiopathic non-pressure hydrocephalus patients. And these IMPH patients come to the clinics because they start to observe some memory impairments, incontinency, and problems in gait. And if they are diagnosed with IMPH, they are treated by insertion of a shunt into the lateral ventricle to bypass the accumulating CSF. And during that shunt surgery, a piece of brain is obtained for diagnostic purposes, and studies have shown that actually if you stain these biopsies for Alzheimer's disease pathological hallmarks, approximately 40% of the patients have A-beta deposition, and additional 10% of the patients have also tau. And actually, but these patients don't yet have clinical Alzheimer's disease. However, if you follow these patients for, let's say, 5 to 10 years from the surgery, eventually 25% of these patients that have pathology will eventually develop also clinical Alzheimer's disease. So we actually have a window to the brain to evaluate in these living brain biopsies the neuronal network functions and operational properties and how early AD-related pathology alters those.
Wow, sounds fascinating. And what kind of collaborations do you have both within and beyond the university?
So we naturally collaborate a lot with the clinicians. So this collaboration is super tight. We have regular meetings and draw from their expertise. We also collaborate within the university. We collaborate a lot outside Finland with top-tier research groups, and we also collaborate with companies and, for example, patient organizations.
Nice. And what are some of the achievements of your research group that you are proud of?
So maybe not mentioning here scientific achievements, but I'm mostly proud of the interdisciplinary and well-functioning group that I have built. The people in my lab are definitely experts in their field. I'm really, really proud of the collaborative atmosphere that has really enabled us to set the frameworks for example, the use of these human biopsies. And I'm really proud of the collaborative pipelines that we have that involve both the clinicians and other research group at the university. And these pipelines are so super important in efficient utilization of these very valuable brain biopsies.
True. And if you look into the future, what kind of impact would you like your group's research to have in the next, let's say, 5 to 10 years?
So I foresee that within the research that we're doing, we're able to pinpoint novel possible therapeutic targets of Alzheimer's disease. Maybe even pinpoint some of the some early AD predictive biomarkers, and really understand the mechanisms, for example, underlying deficient synaptic plasticity in Alzheimer's disease.
Wow, sounds very impactful indeed. Then the most important question: why should researchers choose your research group for their postdoc? What makes your research group so amazing?
Yeah, maybe I could here again refer to the wonderful collaborative atmosphere that we have in the lab. We have experts. I mean, my lab is really interdisciplinary. We have experts in electrophysiology. We have experts in stem cell modeling. We have experts in state-of-the-art wet lab techniques, and we also have a computational arm in my lab who are developing these approaches how we can really do this integrative data analysis and prediction of the cellular responses.
Wow.
So mainly this one. So and of course these atmosphere in which we're anybody coming to the lab will be exposed to collaborations with the clinicians as well as collaboration with the companies.
Wow, sounds amazing. Thank you very much, Tarja Malm.